John and I are both sitting in the building that's shown there, the Venner Institute here in La Jolla. And of course, we're in different rooms because of the pandemic and so forth. And I think I got this last spot because I I'm averse to getting up very early. But however, I got the last spot on that pretext, and then I got up at 4 and came in here for the meeting at 5. So if I'm a bit groggy, please cut me a bit of slack. Okay, so the slide isn't advancing. Okay, we've been driven by this question, what is life? And when I say what is life, I mean it in the sense that was, I think, first enunciated by Schrodinger in the 1940s, and he posed the question, how can the events in space and time which take place within the spatial boundary of a living organism be accounted for by physics and chemistry? Okay, so fast forwarding whatever it is, 80 years or something, we put this question in the context of modern genomics. And when we view the question, what is life, through the lens of genomics, we convert it to what genes are necessary for life, what are their molecular functions, and how do they work together to produce life. So we got into this in the mid 1990s, and looking at the organism M. genitalium because it had the smallest genome known at the time, although it wasn't precisely known. So we helped with sequencing that genome. And over the years, we've evolved the system so that we have a design build test cycle for building chromosomes. And we made SYN 3.0, which is smaller than any other known autonomously replicating cell. So in describing SYN 3.0 5 years ago now, quite a while, we classified the annotations of the genes into 5 categories, ranging from unknown, completely unknown, to equivalog, which is pretty certain. And unfortunately, or surprisingly at the time, about a third of the genes were not assignable a specific function at that time. And why? Are there totally new unknown essential functions? I don't think we can still answer that question. And to put this in context, even though there are 150 or so genes that don't have a clearly defined function, the situation is much better here for a complete description than on other known cells. For example, in E. coli, about a third of the genes don't have described functions, and that's 1,600 genes, not just 149. So there've been a number of groups have looked at this question of identifying the functions of those 149 genes. And we've looked at it, in particular, Chuck Merriman has looked at this, and it's kind of going through the actual annotation is not that fun to do. So let's just talk about the bottom line. So we had 149 genes of undefined function in 2016, and now it's down to about 100, mostly on the basis of bioinformatics combined with experiments published on not really the synthetic cell genes, not really mycoplasma genes, but other kinds of mycoplasmas or related genes in other species. And so now we're down to about one, if you just look at the completely unknown genes, about 1 in 8 of the genes in SYN30 is completely unknown. And we can draw a picture of the cell and label it with the names of processes that are genetically controlled. And one way to look at the annotation improvements is I boxed here all the processes where the annotation improvement has added some new gene to the system. And it's a lot of the processes, as you can see. So it's getting better, but it's still, there are enough genes of undefined function that it's hard to know whether there could be some completely novel processes going on. And the genes that are unknown have diverged from whatever their ancestors were so rapidly because the mycoplasmas mutate fast. They've diverged so much that they aren't identifiable with genes in any other species. So we're now into a collaboration with the Zan Luthi Schulten group and the people in that group to understand the cell by looking at computational models. And the main idea here is that if we can build an accurate predictive model, including all the cell components, it probably does mean that we, in some real sense, understand the cell in terms of the laws of physics. And we can then be in a position to convert the question, what is life, to a emphatic what life is, or life what it is, I guess you would say these days. Okay, so our goals then are to have a cell where every gene is needed and we know the function of every gene. And we're essentially at that point, but now people in Zan's group and possibly others are working on predictive models of the cell. So that will show what the necessary components are and how do they interact. And we also are interested in seeking a description of the cell in terms of a set of universally required tasks, because sometimes a different set of genes could perform tasks. So the gene set is less universal than the task description, I think. And as you've heard at this meeting, that the description of the cell by computer model will provide a design tool which will look at consequences of adding new genes. So with that, I think I'll stop and ask if there are questions.
Clyde, you and I talked about the remaining unknown genes, how many of them, almost all of them, are at least using a simple BLAST-P or PFAM search, says that they are unique to the Mycoplasma mycoides group.
Right. I mean, which that was kind of what I was alluding to when I said these genes mutate very fast. So they're, I think, not it's hard to evaluate what that means in terms of are there novel genes there, because they could be genes that are present in other bacteria, but they've just mutated beyond a point of recognition in mycoides.
You know, you and I still think it is possible that there are cellular functions that are being performed in our cell that we are completely unaware of.
Yeah, I think that's possible. I feel when we published the paper in 2016, we thought that was very likely. Since so many of the genes we identify fit into the known picture, I'm less convinced that that's true.
Mm-hmm. So, Clyde, maybe just
Zan, can you identify yourself, please?
Yeah, this is Zan Luthi-Schulten from the University of Illinois. I'm one of the collaborators listed at the end of Clyde's slides, and my group is trying to do the computational modeling of this minimal cell. So we do know from the proteomics and the essentiality that about a third of those unknown genes seem to be essential. And that was from the essentiality tests that were done, you know, in the eLife paper in 2019. And there are a few transporters that have a reasonable proteomics numbers, but you one time said something to me, if I take the transporters out, that can you give any prediction of how many of those other 30% will you think that they will be involved in metabolism? Somehow you felt, I think it was more of a gut reaction, that it was somewhat unlikely. Do you have any further thought?
Probably was no basis for
Okay.
a gut reaction.
Okay, so we'll just have to keep testing. And I think probably one of the nicest way, fortunately I had to miss most of the talks because I teach Monday morning and I have office hours. So I heard you guys were running late, so I ran home to get on them on the connection. But I think somebody from your group must have talked, or maybe it was James, about how you used so effectively this CRISPR-I technique just to turn off some of the genes.
Was Yo Suzuki, but yes.
Yeah. So has there been any, you know, advancements from that of trying to look in at least those 30 that seem to be essential? If he turns off those one at a time, can we learn something? I mean, that was effective when you guys were looking at SYN3A and realized that some of the 19 genes you had put back in, you really didn't need to put them back in, right? Have you looked at more of those using that technique?
Are you on the
Uh,
line?
So still, making progress on that front.
Uh-huh. Okay. Yeah, no, as I said, the list of the essential genes in that unknown classification, I can resend that to you. It's in one of our tables, but that's the trouble with these computational models. The grows and grows and grows, and because it becomes increasingly important way to communicate.
Zan, so certainly a number of the unknowns are likely to be involved in metabolism in the sense that they are protecting the cell from byproducts caused by sloppy enzymes.
Yeah.
But
I
anyway,
mean, but well, John, you talked about that recent work on looking at the metabolites that are, or reactions that can involve metabolite damage and repair. And some of them classifications contained possible enzymatic reactions. Is that far enough along that one can start seeing where that is? I just remember there were a lot clustered in central metabolism.
yes.
Do you have any sort of key predictions there?
Well, so the work done by Valerie Creasy-Legard, Andrew
Right.
Hansen, and Chris Henry, Oliver Fine's groups, and our groups suggest that some of the, the roles of some of the hydrolases, and there were some other proteins that they speculated about in the manuscript that hopefully is about to go in for publication. But that's about all I know now.
Yeah, no, we took that to heart because one of those hydrolases was a phosphatase that seemed to play. Their mass spec information through knockout studies really showed that they could be affecting some of the nucleotide and the deoxynucleotide formation. So we took that as fairly well grounded based on the mass spec as well as the infinity. But I wondered if one had looked deeper into the central metabolism.
Not that I know of. Not that I know of.